Now if you just take a look on this diagram, here what happens, I have shown the plant cell. Now the cell wall is made up of cellulose. Cellulose are microfibrils. These microfibrils have cellulose threads. All these threads are combined, twisted to form microfibrils, which are again twisted to form in a network structure which we call as cellulose present in the cell wall. But basically these cellulose molecules are having the repeating units of glucose only, where beta 1,4 glycosidic linkage is present. Okay, so I think cellulose will be clear to everyone. Moving further, the next biomolecule which we are going to discuss is chitin. Have you heard about chitin somewhere? Yes, chitin is found in fungi and in some algae and also in the exoskeleton of insects. When you were studying about the diversity in bio, you must have come across chitin. They have chitinous exoskeleton. That is because of the chitin present. Chitin next to cellulose, it is the most abundant polysaccharide in nature. Why it is most abundant? Because it is forming the exoskeleton, and although see, you can see the possibility where they can be found. They are found in the exoskeleton of whole insect group, and they can be found in algae also and fungi also. So they have a um, wide range of organisms where they can be found. So after cellulose, what is the most abundant polysaccharide in nature? It is chitin. Now coming on to the structure, just have a look here. You can see this is N-acetyl glucosamine. This you have to understand and remember that here the repeating units are N-acetyl glucosamine, and if you can see here, the type of repeating units are same, so it is also a homopolysaccharide. Clear? And I have already told you where they are found. They have excellent, because of this complexity they show, that is beta 1,4 glycosidic bond, they have extremely good property of having mechanical strength. They show very good mechanical strength. That is why they can act as exoskeleton also. Just correlate and build up your concept, children. So because it is forming exoskeleton, so ultimately it has to be water insoluble, and this is the property. It is water insoluble, it has tremendous mechanical strength, and the type of bond present here will be, again you can see, it is beta 1,4 glycosidic linkage. You need to understand the structure, that here the carbon has been replaced by a nitrogen group, amine group is being added, which is again bond to a ketone group. Here carbon is directly bond with the double bond to oxygen, and another carbon group is added on the other hand. So this is a ketone group which is added to nitrogen in between. So this is N-acetyl. This whole group is acetyl group. Acetyl group is added, and the portion is called as glucosamine, because glucose 6 carbons are present, amine is present, and acetyl group is present. So this is glucose, amine and acetyl. So N-acetyl glucosamine is the name. In short form, we write down it and in capitals of NAG, NAG, NAG in a sequence wise. But you will draw this diagram structure for this structure is very important for chitin. See, whatever structures I am telling you to learn, just practice one or two times, and you will be able to understand. It's very easy. You know the glucose structure now. Now just add a nitrogen. Amine group is added on the second carbon, and then it is attached to the acetyl group, that is a ketone. Now here it becomes very important to understand and practice the diagrams, because anytime I may ask you the diagrams, the structures also. Okay, so moving further, see the sources of chitin. I have made a collage for the pictures of all these insects which can have the exoskeleton which is made up of chitin. Okay, next uh, biomolecule which we are discussing, next carbohydrate actually which we are discussing, is peptidoglycan. Some of you must have heard about peptidoglycan, and it is a new, or probably it will be a new this thing carbohydrate for you. It is found in the cell wall of certain bacteria. So it is very important for you to understand the importance of peptidoglycan, because we as biotechnologists are very much uh, involved, very much interested in the bacterial origin, in the structure of bacteria, because we need to culture bacteria every now and then in the laboratory. So we need to understand what is the cell wall made up of. So the cell wall in most of the bacteria, most of the gram positive and gram negative bacteria, I'll tell you about gram, what is, how is gram positive, gram negative differ, and what is the basic thing. This will be doing in practicals. So Gram is the name of the scientist who has differentiated between the types of the bacteria on the basis of their cell wall composition. Okay, so this cell wall of most bacteria are made up of peptidoglycan, and this peptidoglycan is giving them this unique property to sustain, to resist the environmental changes and remain alive, or to form a capsule, if you remember the capsule structure. Now coming on to the repeating units. Peptidoglycan is the, see, can you see the repeating unit? It is N-acetyl glucosamine and N-acetyl muramic acid. I have written only the short forms. Here you no need to understand, no need to learn the structures of N-acetyl muramic acid, but you know already know the structure of N-acetyl glucosamine. So glucosamine till glucosamine is okay. Now the repeating unit will be alternate. N-acetyl glucosamine linked with N-acetyl muramic acid, N-acetyl glucosamine linked with N-acetyl muramic acid. This is how the chain goes. So bacteria attain this proper shape and retain environmental condition due to presence of this peptidoglycan biomolecule. Clear? So what is the type? If you can see that the repeating units are alternate, so what will be the type of what will be the type of the polysaccharide, which is this? Yes, this is heteropolysaccharide. Why? Because the repeating units are alternate. They are not of same kind. Clear? Now, they also have, these polysaccharides are also cross-linked by peptide cross-links, peptide bonds. These kind of muramic acid bonds are cross-linked also there. So protein can also be present in between, so form such kind of complexity. So it forms a complex structure of peptidoglycan. We are not discussing the structure in detail because it is not given in your syllabus. Just you need to make the small chunks having NAG, NAM, NAG, ANAM in alternate manner. Remember to draw it in alternate manner. See the gram positive and gram negative bacteria. The red colored coating is the thick peptidoglycan. The gram positive have thick peptidoglycan layer present, whereas in gram negative, the gram staining, Gram was a scientist who developed this kind of staining method. That is why we called as grams positive, gram negative, because those who are giving color are gram positive, those who are not giving color are gram negative. You will come to understand this is a method of differentiation between the different types of bacteria. Actually bacterial cell has been removed from your syllabus, so we cannot discuss it in detail. Now this you can see the red color here. The red color one is very thick in gram positive, thick peptidoglycan, but here in the gram negative one, it is just a thin layer as compared to gram positive. So this is the structure. You just have an overlook, no need to study this structure in detail.